Chapter 18, Body Fluid and Circulation. You have learned that all living cells have to be provided with nutrient, oxygen, and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have effective mechanism for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for this transport. Simple organisms like sponges and coelacanths circulate water from their surrounding through their body cavity to facilitate the cells to exchange these substances. More complex organisms use special fluid within their body to transport such materials. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also help in the transport of certain substances. In this chapter, you will learn about the composition and property of blood and lymph, tissue fluid, and the mechanism of circulation of blood is also explained herein. 18.1 Blood. Blood is a special connective tissue consisting of fluid matrix, plasma, and formed elements. 18.1.1 Plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water, and the proteins contribute 6 to 8% of it. Fibrinogen, globulins, and albumins are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanism of the body, and the albumins help in osmotic balance. Plasma also contains small amount of minerals like Na+, Ca2+, Mg2+, SCO3 negative, Cl negative, etc. Glucose, amino acids, lipids, etc. are also present in the plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without clotting factors is called serum. 18.1.2 Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements. Figure 18.1. And they constitute nearly 45% of the blood. Erythrocytes or red blood cells (RBC) are the most abundant of all the cells in blood. A healthy adult man has on an average 5 million to 5.5 millions of RBCs per millimeter cube of blood. RBCs are formed in the red bone marrow in the adult. RBCs are devoid of nucleus in most of the mammals and are biconcave in shape. They have a red colored iron containing complex protein called hemoglobin, hence the color and the name of these cells. A healthy individual has 12 to 16 gram of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, graveyard of RBCs. Leukocytes are also known as white blood cells, WBC, as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which averages 6,000 to 8,000 per millimeter cube of blood. Leukocytes are generally short-lived. We have two main categories of WBCs: granulocytes and agranulocytes. Neutrophils, eosinophils, and basophils are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells, 60 to 65% of the total WBCs, and basophils are the least, 0.5 to 1% among them. Neutrophils and monocytes, 6 to 8%, are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Eosinophils 2 to 3% resist infection and associated with allergic reaction. Lymphocytes 20 to 25% are of two major types, B and T forms. Both B and T lymphocytes are responsible for immune responses of the body. Platelets, also called thrombocytes, are cell fragments produced from megakaryocytes, special cell in the bone marrow. Blood normally contains 1.5 lakh to 3.5 lakh platelets per millimeter cube. Platelets can release a variety of substances, most of which are involved in coagulation or clotting of blood. A reduction in their number can lead to clotting disorder, which will lead to excessive loss of blood from the body. 18.1.3 Blood groups. As you know, blood of the human being differ in certain aspects, though it appear to be similar. Various types of grouping of blood has been done. Two such grouping, the ABO and RH, are widely used all over the world. 18.1.3.1 ABO grouping. ABO grouping is based on the presence or absence of two surface antigens, chemicals that can induce immune response, on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibody proteins produced in the response of antigen. The distribution of antigens and antibodies in the four groups of the blood A, B, AB and O are given in table 18.1. You probably know that during blood transfusion, any blood can't be used. The blood of a donor has to be carefully matched with the blood of the recipient before any blood transfusion to avoid severe problems of clumping, destruction of RBC. The donor's compatibility is also shown in the table 18.1. From the above mentioned table, it is evident that group O blood can be donated to persons with any other blood group, and hence O group individuals are called universal donors. Person with AB group can accept blood from persons with AB as well as the other groups of the blood. Therefore, such persons are called universal recipients. 18.1.3.2 RH grouping. Another antigen, the RH antigen, similar to one present in rhesus monkeys, hence RH, is also observed on the surface of RBCs of majority, nearly 80% of humans. Such individuals are called RH positive, and those in whom this antigen is absent are called RH negative. An RH negative person, if exposed to RH positive blood, will form specific antibodies against the RH antigen. Therefore, RH group should also be matched before transfusion. A special case of RH incompatibility mismatching has been observed between the RH negative blood of a pregnant mother with RH positive blood of the fetus. RH antigen of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to a small amount of the RH positive blood from the fetus. In such cases, the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, the RH antibody from the mother, RH negative, can leak into the blood of the fetus, RH positive, and destroy the fetus RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This can be avoided by administering anti-RH antibody to the mother immediately after the delivery of first child. 18.1.4 Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to any injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at the site of a cut or an injury over a period of time. It is a clot or coagulum formed mainly of a network of thread called fibrin, in which dead and damaged formed elements of the blood are trapped. Fibrin are formed by the conversion of inactive fibrinogen in the plasma by the enzyme thrombin. Thrombin in turn are formed from another inactive substance present in the plasma called prothrombin. An enzyme complex, thrombokinase, is required for the above reaction. This complex is formed by a series of linked enzymic reactions, cascade process, involving a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by the tissue at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting.